Good morning, Ty. Hello, hello. That. 2 cans of clean. Share materials today. Good morning. Welcome. Try. Put up to you. Well, we're gonna get it or we're gonna get back later. The way it works. Yeah. This is great. We're all gonna get it. 200,000 cases and 2,700 deaths yesterday. Unbelievable. Unbelievable.
Is this,
They've all been.
what's the yellow cube?
I honestly believe we're starting to stain the desks with the yellow solution, which I'm sure nobody thought of before we started using it. But you know, good stuff, right? Everybody, so we can get everybody in the same place at the same time. Please log into those Pear Deck slides. We're going to be using those if you're learning from home. Less so if you're in the building. Alrighty. All right, see if we can get this to work. Folks at home, are you willing to say hi to the folks in the classroom?
Hello.
Maybe.
Fair enough. And folks in the classroom, are you willing to say hi to the people at home?
Couple of technology.
Oh well,
issues.
Catherine is at home now. I didn't know that.
You're having technology issues.
Gotcha. Sorry. All right. Sorry, Mr. Peck just stopped in saying that people were having technology issues. So, all right. So we're going to try something different. And it's worked so far pretty well. So I'm hoping it'll work well for you. So I've got two different lab experiences, but really it's the same lab experience. One is for people here in the classroom and one is for people at home, okay? So I'm actually, just for the sake of time, I think this makes sense unless anybody objects vociferously. We'll skip the brief check-in. We'll move it to next time. And what you want to do, I'll put you in student mode so that you can actually do it, is click on the link that matches where you are right now. Okay. So the code is right here. Sure. It's always in the upper right-hand corner, but I think the picture of me now is blocking it. So just make sure that everybody can get to the right place before we get started. Okay. Just doing okay? Sorry, the code is hiding behind my beautiful mug. Sorry.
What was that last one?
F.
Thank you.
You're welcome. Okay.
Wait, which slide? Or are you gonna put it on?
Ideally, it will be up here, but if you want to pull it up on your device, all you should have to do is click on the link and it should bring you right to the slide.
I think we're a student.
No, we ended up on the third slide, so now we have to like go all the way through.
Oh, okay. So
Is this in
let's just do this then.
There we go.
I just don't know that you'll be able to click on the link, but I'll tell you that if you're in person, just click 2 slides forward and it's going to be what you want. I was trying to build in a shortcut, but if it's more work, it's not worth it. Okay. All right. Doing okay? Thumbs up. Folks at home, thumbs up. All right. So let me just ground everybody in what we're going to try to accomplish, no matter where you are today. Last time we looked at hydrolysis reactions, right? They're the type of biological reaction that's responsible for catabolism. Okay. So we want to use a really simple chemical reaction to verify that hydrolysis is actually a thing, that it's actually possible. So the experiment that you're going to set up if you're in the classroom, which is really similar to the virtual experiment that you're going to perform at home, uses 2 different sugars, sucrose and glucose. Okay, remember, sucrose is a disaccharide. It's composed of glucose and fructose. Glucose is a monosaccharide. It's just a simple sugar. It's a single molecule. Okay, so really what we're asking is, can we hydrolyze sucrose? Glucose you can't hydrolyze because you can't break it down. It's a single molecule. So what we're going to be doing is using a really simple biochemical test. It's called a Benedict's test that indicates the presence of glucose. And the mechanism of that reaction is way outside the scope of this class. But suffice it to say that if glucose is present, Benedict's solution will react with it in a concentration-dependent manner. So the more glucose that is present, the darker, or I guess more red would be a better way to say that, the solution will become. Okay. But the glucose will only be present if the sucrose gets hydrolyzed. Benedict's solution will not react with sucrose. It will only react with glucose. Okay. So if we can successfully hydrolyze sucrose and release the glucose from the disaccharide structure, we should be able to verify its presence with this Benedict's test.
Do you have to like measure or anything?
Nope. Nope, this is all qualitative today. Because literally we have to find out if this system is even going to work in the first place. Not the reaction. I know the reaction works. The experiment is going to work. That's not what's in question. Can we actually do labs in the classroom while folks at home are doing something similar wherever they are? Go ahead. Sorry. Right? So folks at home, you can click on that link, or you can go to Google Classroom and just find lab number 8, and you can get started, okay? And if you need me for any reason, the speaker is on. Just say my name, and I'll come back to the computer. For the people here in the classroom, I'm going to move over and get them started with their experience. Should we give it a shot? All right. Back a little bit. All right, so folks here in the classroom, you just want to find a lab station. And let's get it spread out as we can. Okay, so there's 2 kind of behind Jessica, and then there's 4 behind Tenley, and then there's 4 more behind.
Do we need computers or?
This will be up on the screen, but if you want a closer up view, you can bring your device. Let's go. Let's do it. Let's try it. We got to find out if this is going to work.
This is regular water.
It's flowing water. Might add some more too. So, all right. Can I bring it? We'll have it if I bring it.
Feels good.
All right. So I want to do one thing just before we get started because I wasn't sure if you've used size headers like this before. Is it familiar to you? Yes. So people say yes, people say no. So if you've never used one of these before, I've given you 3 5-mil glass pipettes. What you want to do is just insert the blunt end, not the point end of the pipette, into the pipe header. Just push it in far enough that it stays. If you push it in too far, it actually break off in your hand. You don't want it out. Okay. So if it's secure, you should be good. And if you look at the pipette, it's kind of a funky measurement scheme. It starts at 4 and goes up to 0. Okay, but if you spin the pipette a little bit, you'll see that the 0 is 5 milliliters. Don't get confused. The 0 is not 0 milliliters. It's 5 milliliters. It's like you're counting backwards. So if you see the 4, that's 1 milliliter. The 3 is 2 milliliters. So one of the measurements you're going to have to make is 3 milliliters. So, you know, I could use either the glucose solution or the fructose solution or water, but since it's clear, it's going to be kind of hard to see from where you are. So I'm just going to use the blue Benedict solution as the example. You're not going to measure 3 mils of Benedict solution for this, but that way you'll be able to see it from wherever you are. Okay, so it's with the pipette in whatever your dominant hand is, the little wheel at the top. Put your pipette in. Spin that wheel down, it'll bring the solution up. Just like measuring any other liquid, find the meniscus, and that'll be your measurement. When you're ready, spin the wheel the other direction, and the solution is expelled from the pipette. Just like that.
Does it matter if there's water in the pipette right now?
Okay, that will work. Precision. This is qualitative. So what you want to do is just use that chart, whether it's in front of you or up on the smartboard, to set up the 3 tubes with the volumes as listed. And those 3 tubes will go into the boiling water bath for 3 minutes. We'll take them out, we'll add the Benedict solution, which is 5 drops, and then we'll see what there is.
And use a different pipette for each substance.
Unless you want to rinse it out in between. So I gave it to you. Speed things up a bit. Should have water in those speakers too. small numbers, we use a small. They're gonna count up to 5.
Sucrose.
I already said so.
Well, I said it isn't.
The good thing about biology is pretty much everything is edible. The question is, what is it gonna do if you want to eat it?
What would it to you? What happens? Taste good? Doesn't it taste like sugar? Isn't it just sugar?
It's sugar solution.
Did you make it?
I think it's, I think I might sugar. You could drink it, it tastes sweet. My philosophy is simple. You can taste anything in biology lab. It's just less for me to grade. That's how it works for me. If you do me a favor.
Heck with this list, dude. Quick look at HCL.
Everyone at home doing okay? Finding what you need to find?
Which one is, how much, this solution?
The Bendix goes in after the samples are heated into one cell. It'll be 5 drops.
Where's the which one? How do I hold it there?
So it's coming out, it means it's pipette, it's secure, push it a little bit more tight. It might be on the other side, it might have gotten moved.
Is HCL the Benedict's or is the other one?
The HCl is in the little dropper bottles with the red tape on them.
This is
Water.
don't taste that stuff.
It's still dilute. I mean, it's interesting because somehow along the way, like, I don't know, you guys are super curious about what these things do to you. But I think as teachers, we've created like this sense of fear in you. Like, if you do this, you're gonna die, or you're gonna melt or burn. You're carrying hydrochloric acid inside of you right now.
But not on the outside.
No, not on the outside, but you know, if it were to find a way to leak out of your stomach, there would be some pretty significant problems.
Isn't that like when you die, acid like slowly deteriorates in your insides?
Oh, sure. I mean, if you're not replacing the cells and the mucus that line the inner portion of your stomach, oh yeah, eventually it'll degrade. Another way to make glasses. Run an experiment where, through human decomposition, you allow yourself to dissolve from the inside out.
Okay, then we put these in the water.
All 3 go in the water bath.
Can we take the HCL straight from this if we just use this on the still water?
Well, you're just gonna add drops of it.
5 drops.
Yeah, so it's like, once you open the lid, you're gonna see the little dropper.
Should we set them in or hold them?
Some people have had some success just setting them in, but if you feel like they're gonna slip, you can just hold them. It's only 3 minutes.
The dropper is supposed to be like part of the time. Oh, it's like this. Where's the timer?
Never done it before, you can't be stupid. At this point in the year, we would have probably done like 10 different labs. This is the first one.
I wish we had made more.
Well, we can keep in mind, we did you, what, 7 virtual labs, right?
Yeah, those aren't.
I know, but I tried. I tried.
You just broke Mr. Bergeron's heart. Why didn't, they weren't bad. They were like. 10.
They were kind of fun. I
They
mean,
were like 5%. They're like 5%. Incubate them for 3 months.
Mr. Bergeron, why is this like?
Wait, is it, are they supposed to just be able to rest?
hopefully, yes. Yes. If you feel like it's not stable, though, you can hold it.
Can we, I assume we can sit on these, right?
That's one, we've got to just begin to understand this is a respiratory disease with particles that are in the air. I cannot sterilize every surface in the classroom.
Pushed it in there, it's still like, it's always.
I mean, aren't the odds of like getting it from touching something like less than, a lot less than like.
You have to touch something, right? And then touch your eyes, your nose, or your mouth. And really, you have to touch most likely your nose or your eyes.
Unless if I roll my eyes right now, I'm getting
I don't know if that's true. I don't know what the viral concentration is in the classroom right now.
Mr. Bergeron,
Good job.
Yes,
Mr.
sir.
Bergeron, I'm confused of what the positive control is for the lab.
So remember, it's been a little while, so it's understandable. Remember, a positive control is a part of the experiment where you expect a change in a dependent variable. So these are qualitative experiments, right? We're looking for a color change. We're not measuring the extent of the color change, really. So is there a tube or a part of the experiment where you expect the Benedict solution to react and cause a color change to occur?
Me, Jonah, and
When you add like a bunch of, glucose, yes.
Yeah, because glucose should react with the Benedict solution. There should be a change in color.
And the negative control would all just be sucrose?
Yep.
Without the HCl. So
Right.
one, okay.
You got it.
All right, got it.
so I got like a. I don't have any errors now. I just use the ones I find. But I would like to build mine. So I like these. What is the HCl do?
Mystery.
HCl plus sucrose. Oh, I think I need more Benedict solution. Yeah. We use the pipette to get the solution.
No, there's a little transfer pipette. The plastic. Yeah.
Somebody, like Mr. Bergeron. This one can like pull it off of the state.
Can we move the test tubes, the hot test tubes back to the thing?
Is the DH2O? Oh, distilled water.
Try something else.
I forgot which tube was which.
Here, college admission process. And let's tell you a little bit about.
It's the like,
Let's get that one. Start taking them out. Circulation.
Wait, how am I?
Okay.
Hey, one of them turning green. Oh, 2 of them are turning green. This may need CL. That's cool.
Wait, Mr. Bergeron?
Uh-huh.
Who was supposed to be there?
Tube one should be, it might be a little blue, should not be orange.
Bro, let me see.
glucose or sucrose? You sure that's
I don't have that. I don't like it. So, like this one has the griddle.
I care less about it without understanding.
Maybe it looks slow.
Why
I
we don't pick with it.
don't want to be compared. Is this your Bergeron?
Mr. Bergeron.
Does this look like I messed up?
It's an experiment, you can't mess it up.
No, like, if I got all the volume correct, this is a dollar roll.
Mr. Bergeron.
Oh, that's not-
You just can't win. You can't win.
Oh, that's good. So all I gotta do is take a picture and then submit it.
There's a very short write-up.
Label your image and submit your work. Is this the lab for the week or is there another one on?
There's a, in classroom, there's a handout.
How much Benedict solution do we have? 5 drops.
Yeah.
How do you drop it from this?
You guys look like that.
Little like that.
Look like that. Transfer pipette, plastic pipette.
I wanna go see Mr.
So if you have finished, you can clean everything up. Or they can go down the sink and toss it.
What does boiling it do?
What do you think it does?
Water can't go in from the top.
Sorry?
It just, yeah, I guess it just, yeah. I mean, how is it gonna get hydrolyzed from sucrose to glucose? It's 2 to 1. That's like dye to mine. So then you would add water to it. No, sucrose to glucose, you take water out.
Where's the first time?
Well, when it turns into glucose and fructose, water is about. Oh, boy.
That's dehydration.
Wait, what? Wait.
Hydrolysis consumes water.
Sucrose is the big one. So then it turns into 2 small ones. Oh, that makes sense. The big one has, when you turn it into the 2 small ones, water comes out of the 2.
It goes in, it breaks the bond between the two, and then becomes parts of the 2 molecules that are produced as products.
Wait.
No, the guy with 2 master's degrees is telling you a lot.
Okay, wait. So it starts big and then water goes in. And that's called getting it like hydrolyzed. And then the one, like the OH will be 1 and the H will be set to 1.
And so then I did a retest and I put in the retest and I office, that was for the semester.
Well, probably should watch that.
Like, what's doing to me, that's not what I got out of it.
That's not a bad idea, that's why they're there.
I just did like the correction.
I took notes.
We need to take a picture of this for classroom?
You can, yes.
One of my characters contaminated.
There is a sample image up on the document that you'll be finishing.
But I mean, the steam can't go in through the top. It's not like the water's being added that way. Is that why we're adding boiling water?
No, there's water everywhere, right? There's, the solutions are made with water.
Oh, yeah.
The boiling water
That's
bath
good.
is,
Well, then why does sucrose get hydrolyzed just like by itself?
that's a great question, right? There's a lot of heat there, a lot of thermal energy.
Oh, because it's really endothermic or exothermic.
I got a C, I didn't study.
It's just got a high reaction activation.
Ooh, now we're getting closer.
Well, because you had to add it to boiling water for it to do it.
But in 2-1, you had sucrose, and those samples went into the boiling water. Was there a reaction?
There was.
Well, yes. So I think that's because of contamination, not because of the actual reaction.
I don't know how they're contaminated. Maybe the process.
This is a good time to clean everything out for the next.
Wait, what is the thing? Mr. Bergeron?
Not a Benedict yet.
Huh?
You're not a Benedict yet.
Is this like the whole
Add
lab?
a Benedict solution after the heat come out of the water.
Wait, this is lab number 8, right? The blue is this week.
Should we don't do that?
Once you're done,
I have a
clean
question.
everything out.
My sister ever called you Bird there?
I don't think so.
Okay,
Which
but that's what she
sister?
called you, Anna? called you that, like at home, she called you bird bear, but I don't know what she ever called you that in class.
I'm pretty sure Liza never called me that, no, but Anna?
That was Anna, that's what Anna
I
called
feel
me.
like Anna might have had nicknames, I don't remember.
Can we dump these in the sink?
Yep, it's all water soluble. Or I'm gonna have water soluble for long term, not toxic.
The mine especially. Oh my god.
Rinse everything out really well.
Dump it in the bowl. Oh yeah, what is this copper? Is that what it's? Copper, right? I didn't know something about biology. What?
Mr. Bergeron, there's like all that stuff that on TV.
If you rinse them out with warm water, they might stain a little bit. but-
Warm water?
Yeah, it might stain a lot, it's okay.
Okay.
What?
I know, like,
Call Abington.
You guys know.
But if you have finished, there are some activities, they're linked on the agenda, that give you a chance to review fatty acid structure and function. I would encourage you to take a look.
Still nothing's happened.
Have you added the Benedict solution? If you add it after they come out of the water bath, they're still in the water bath.
To add the Benedict solution to the water heater. So how did you expect the Benedict solution to react? Since there's a glass barrier. Just like out of curiosity.
It's a 5 to each. Dude, the water is shocked. Water is shocked.
Yeah. The Stuber water fountain used to shock me when I was drinking something else, like through the water. We had to rinse the pipettes.
The glass one, yeah, the plastic one is fine, we'll use it for one thing.
I mean, if I organize them so that the group knows like which use on which.
Just rinse.
I mean, mine was gonna take me probably.
Just rinse everything. It takes 30 seconds.
How do you do it though? So, in the sink?
And then after we rinse all that, we're good and there's activities.
Yes, so you hopefully watched the lipid fatty acid video, there's a couple of activities linked
Yeah, I
on the
did, was that
agenda.
one that auto turned in, because I watched it on my phone and it didn't auto turn in on Classroom, so I marked it as done.
That's fine, I check Edpuzzle for completion, so you
Okay,
can.
fine.
You're.
There's plenty of things.
Do we need to be in student mode to get to the other?
It would probably make things a little bit faster for you unless you wanted to go to Google Classroom.
It's like a third course is like.
So I thought this was maybe like.
160 something?
Oh.
No, anyway. We got a lot of slides. Is this an entire semester worth?
Oh, yeah, I don't
No,
know.
this is the unit that I'm,
It's snap.
I don't even know how to describe it. I create stuff on the fly now.
It's the second. It's the second.
It's a lot
First one had probably
a lot of work.
And this one has just like,
but
it's
it's worth it.
like 4 feet long and it has like a.
Do we need to watch this live egg puzzle?
No, that was for the people folks at
You did, instead of watching.
Oh, I got it right.
If you put like fiberglass highway rods in your fence, it'll make it like really strong. So it has like 8 like fiberglass to get like the taper ring.
Or the links to the activity. You can do that one.
I can draw it back like stuff. It's really cool.
I thought those two would be the ones.
But Anton's dad, let me find his bro. He said it was 65 pounds.
So there's nothing wrong with doing what you're doing.
That was 65 pounds.
Oh, like click on the agenda. Okay.
Yeah, but that practice problem is a good one too.
But maybe Anton's dad. But it's okay.
Oh,
Nicely, appreciate it. So this is the next group.
Mr. Bergeron, how is coronavirus going in like Europe? Was it just as bad as here?
Depends on the country.
Like Britain?
Britain got hammered pretty hard and then they instituted a lockdown. So they're seeing reductions in positivity rate.
What about like France,
France also instituted a lockdown. They were out of control about
Sweden?
4 weeks ago. Sweden's kept it under control for the most part.
Like in Sweden, having the worst because they're trying to like herd immunity and then it's about to be like.
Yeah.
But Germany has like a really low death rate apparently.
I don't know. Germany's different. Yeah, but Tudor's right. Sweden kind of threw all their wood into the fire and hoped that they could hit herd immunity. And, no, in many ways, they were the consequence of probability. They didn't get hit hard early, and then later on, they got hit really hard.
Like, are their leaders smarter than Donald Trump? Well, I know, yes, it's not that hard. But like, is it like as bad as here? Because like right now, we're not good.
Brazil was up there. United States, Brazil,
Yeah, Brazil.
any country that borders on authoritarianism was and is in trouble.
Who's the Brazil guy? Bolsonaro?
Bolsonaro, yeah.
Yeah, he's pretty bad.
Doesn't seem like a nice guy. I've never met him.
And who's the Bolivian president?
Wasn't there like an offensive coup in Bolivia
Yeah.
backed by like the US military?
My dad got a bunch of media attention because he like studies Bolivia. So all the news people are calling.
So my hope is you're practicing with these interactive activities on fatty acid structure. Yep. There's like some multiple choice questions.
Wait, your mom's sister is a conspiracy theorist? Your mom is a conspiracy
There's so
theorist?
much fake news in Romania. It's insane. And they all believe it. There's like
kinda
all these
funny.
sensationalized articles about like American politics.
That's kind of funny.
It's whack.
Everybody at home doing okay? Nothing in the chat. I'm not hearing anything.
Actually, it was just popular, like, it was like massive voter fraud.
We are back on the agenda
Dude,
slide.
there
We're
was.
looking at those interactive activities on lipid
I gotta
structure,
send my
fatty
grandma
acid
some of this
structure.
stuff, 'cause, like the Georgia or their official.
Yeah.
Why is the
We
OH
gave
group
this
the
like
acidic
speech
part?
on otherwise.
Why is the OH group the acidic part?
Other fatty acid.
Yeah.
So carboxyl group, so there's like a superficial answer and then there's a detailed.
It's like there's no voter fraud in
Detail.
So that OH, right? That in and of itself is the O and the H going by.
Oh my god.
The carbonyl group is also polar. Especially a net dipole
in the center.
pulling the electrons towards that oxygen atom that's attached to the carbon. And it weakens that OH bond with one of the H good associates.
Okay.
Which makes the carbon solution more acidic.
Trump is
Okay. That makes sense. I was thinking of it like as the OH group breaking off, which would then be like beyond like basic, but that makes more sense.
Right,
Like,
and then you end up with a carbanion. Like carbon has like
Dude,
a little pair of
Dude,
electrons
he's
sitting
probably,
on the side of
the
body.
reason he's probably fighting it is because he has so much like legal
Thank you.
trouble. Dude. He's already like pardoning like his whole family.
Rudy Giuliani asked for like a preemptive pardon.
Dude, that's super sus. Wait, is this it?
That's the lab write-up document.
We have to fill this out?
Well, we're trying to work on these interactive activities on fatty acid structure. That way, if you have questions, I'm here to support you.
Oh, is that on Pear Deck? What slide?
Seasons total.
The agenda agenda slide for the day, which is 163.
53?
163. But you just search for it, can't you?
You can search for a slide.
Can't you do like like Control F? Even if you just search the agenda, you can cut it down to a dozen slides. Maybe not.
Oh, it was just on student mode. I had like go on. You had to do the sugar and urine.
No, that's what the at-home people did. You did the lab, they watched the lab.
Somebody got better data than me.
Back in my day. I think I discovered that like I think the way it was diagnosed before, yeah, like tests and everything was actually like taste and smell the urine, it was like smell and tasted sweet.
Who did?
Like the doctors who diagnosed diabetes.
Dude, imagine how many diseases. Wait, Mr. Bergeron, what was the role of the hydroxide, the H sign?
I don't know, what do you think?
Well, tube 3 was just sucrose, right? But
With the HCl. So no, it had the H plus ion that was there.
So sucrose is big and you add water. Oh, that does make sense because the little ones each have the water and when you combine to make the big one, it's dehydrated because the water comes out.
Mhm.
Oh, I did watch it right. I was just okay, but the HCl then, with the big one, the big sucrose, which does not have water.
Well, remember, the water is everywhere. All these molecules are dissolved in water.
Well, it's probably, I mean, maybe it reacts, if an H plus is super like polar,
Well, it's an ion, right? It can't be polar.
well, it's charged
Mhm.
positively.
Mhm.
So then maybe the OH minuses in the like sucrose,
But they're not OH minuses, right? They're hydroxyl groups. They're not.
well, are they still negative?
No, they'd only be negatively charged if they were attached to a cation.
So what does the H plus ion do? It's positively, maybe it like makes water, like it forms one part of the water. So like it's initiates the breakup because it already has the H of the OH.
Okay. I think what I would ask you to consider is in tube 1 where there was sucrose but no HCl, was there hydrolysis?
Well, in mine, yes, but it's supposed to be no.
Okay, so no. But I agree, no. In tube 3 where there was sucrose and hydrochloric acid, was there hydrolysis?
Yes.
So it's not really a question of the water, because the water is present in both of those
So
tubes.
it's energy, the activation.
It's gotta have something to do with the energy.
So
Right.
why does hydrochloric acid get more energy?
Did it get more energy?
Give.
Did it give more energy? What might be responsible for a reaction a reaction not occurring in one tube, but a reaction occurring in the other tube?
the energy. Because one didn't get up to the EA.
In fact, it didn't get up to it, right? Because it was too high, which is crazy, right?
Wait, what?
We're boiling the sucrose. And that wasn't enough energy to break the bond between glucose and fructose in sucrose and release the glucose into solution.
Yes.
Right? That's tube 1. But in tube 3, the H plus ion, acting as a catalyst.
Okay.
It's lowering that activation energy barrier. Now there's enough thermal energy in the system to break that
But how
bond.
does it act like a catalyst?
Well, I mean, at some point, that's way outside the scope of a class like this, right?
Oh,
This is
well, that's what I was
intro
trying
life
to-
science.
That's what I was trying to.
Well, but that's a really important point. Like, if you're going to that level, are you asking a question that you think like a typical high school student would be asked to answer.
Well, I didn't know if it was like something like the H plus.
It is. The H plus, you're right. The H plus ion is acting as, it's, you can get into this, into the weeds really, really quickly. It's acting to weaken that bond between the glucose and fructose.
Because it's charged, so then it's like pulling them apart. Kind of.
In a way. I mean, it's not, obviously it can't pull them apart. It's a proton.
It's just making the bond length longer.
You can think of it that way, yeah. Weaker, bond length longer.
So
Assume the hydrochloric acid contains sucrase.
Oh, if only.
What's sucrase? An enzyme.
It's just, you know, but it's just, have you posted everything?
Oh, you're so
with, like, mental break, basically?
Trying to.
bro, what's
There may be some things I need to move around so that I can get back.
Yeah.
And some things are scheduled to appear later.
bro?
So,
I can't get it.
like, I made a summative assessment number 6, but you don't see it yet.
Yeah,
Is there like a separate reaction that the high, like, how else does the catalyst work in chemistry?
Well, biological catalysts, enzymes, work in a way that's similar to what you just described. It weakens bonds in a way that the energy within the system now is able to break that bond.
Like
Before
my
it
body
wouldn't have.
temperature is only 96.
Right.
Well, it's supposed to be, but I got COVID.
That's really important, right? We're still pretty warm creatures, but that still isn't enough thermal energy to allow for a lot of the reactions to occur because the activated energy barrier is still too hot. So while H plus can act as a catalyst in living systems, usually it's enzymes that lower that activation energy barrier and get reactions to occur that otherwise wouldn't.
What molecules are enzymes?
Proteins. That's next week and the week after.
So
And the week after that.
So proteins. Do proteins also react with fats?
They could. They could.
Mr. Bertrand, how should we draw the energy diagram?
You can insert a drawing. Or you can draw your own picture and insert the picture.
Okay.
Whatever is easiest for you.
Look at this.
Thanks for all your work today, folks. We were able to do it successfully.
Watch this. You see how? Hey, but look, the target is. Look, the target is all the way there. No, of course not.
Mr. Bertrand,
I was aiming way too low because if I aim higher, it's probably, I mean, I'd rather. There's a huge space beyond the target, but like after that, it's just like. I made the stupid mistake of like shooting like the. I didn't think it was that powerful since it's only like 20 pounds, so I like shot it at like 45 degrees for like 100 yards. And it went way past the target, all the way to like the top of the tree behind it. But I found the arrow. I kind of want. I have like a huge log of what's it? It's Osage orange. I have a huge Osage orange, and I have a oh, what's that? Like the blade that you use to. I don't know. There's like a YouTube video. It's like a 3-part YouTube video of this guy making a bow from Osage orange. And I have like a huge Osage orange thing, and I would have to like. It's like a 3-month process because you have to steam it and then you have to like let it sit for like 3 months or something. I don't know, bro. You have to steam it and then bend it into the right shape, and then once you do that, you let it sit. I don't know how you make those. You gotta have like one. Thank you, Mr. Bergeron.
Thank you.